all this that I said. For instance, look at this paper. If you knock out the PMAL1, which is the core component of the circadian clock, these mice age prematurely and die early. Also have increased oxidative stress and increased age-associated pathologies. So you knock out a protein on the circadian clock and the animal age, which is quite interesting. Vice versa, if you take a mouse who lives longer, there are many mice models of longevity, but this is the long-lived MUPA mice, which is the urokinase type plasminogen activator transgenic mouse. This mouse, which lives longer, has oscillations of the clock genes that is more accentuated. So it's a better circadian rhythm. So it's the opposite of before. Again, this is the same slide. SIRT1 is not only involved in linking metabolism and circadian clock, but also links these two things with aging. How it does so? So there was a Cell paper last year, Lenny 40. So they created the knockout mice for SIRT1 in the brain, or transgenic mice for SIRT1, always in the brain. And what they saw that SIRT1 can basically, if you overexpress SIRT1, the oscillations of the circadian genes, it's ameliorated, so it's increased, it's the blue line. And also the expression of the genes is increased. Instead, if you have the knockout mice, which is the dotted line, so BSKO, brain-specific knockout, you see that the expression is lower, and not only lower, but for example, look at PER2, this panel here, the oscillations is blocked. It doesn't, the genes don't oscillate anymore without SIRT1. So, and what about humans? So it's very difficult to do circadian studies in humans. I mean, if you want to study aging and circadian, it's not very easy. But this study from Anne Eckert a few years ago, they basically took older people and they took their serum and they exposed cells from young subjects or old subjects. And basically, they want to see if the serum also from the blood from these patients could affect the oscillations of the circadian clock genes. And they found that the serum of the old people make oscillated genes out of phase, no phase or out of phase. So it's like a disruption. And what they hypothesize is one thing. Old people usually are more sensitive to light. So old people like often they don't like being in the light so much because they are very sensitive to light, and the light affects the brain in old people in a different way. And we don't know that in detail, but we know that in response to this, they will secrete hormones, so the endocrine system, which in turn will also affect the circadian clock. So it was very, very early days. I mean, we don't know very much. Okay, to try to conclude a few more slides. So this is what we know more or less as a model. So we have the light and the food. This affects the suprachiasmatic nuclei in the brain. These produce hormones that can affect the peripheral clocks. So I told you these clocks are in every cell, so in the liver and the muscle and the adipose tissue. The nutrient sensing pathways are deeply connected with this. And at the end of the day, we have an effect on our metabolic homeostasis. So when we are hungry, when we sleep, it's all connected. However, what this system, when it's especially the nutrient sensing pathways, it's very important in development and reproduction. But when you have when you age, this system goes wrong. So you are desynchronized. I mean, you all have parents and grandparents. How many hours they sleep? 4 hours? 3 hours? I mean, people need the less sleep usually. They sleep less, and they also sleep worse. They wake up at night several times. That occurs in aging. And in aging, you have also diabetes, you have cancer, you have glucose intolerance, insulin resistance. So the hypothesis that we put forward, many people are working on this, is if you desynchronize the circadian system, this is one of the reasons because people age. And the questions are open questions, like research questions. What are the main factors that determine individual body time? Why I can sleep better than you, for instance? Who knows? Or there are genetic and environmental factors, not in particular. But there are genetic and environmental factors to be quantified. Can we quantify this? I mean, what are the factors? And really how declining expression of clock genes affect metabolism and aging. So I have a project with a group in Brazil. Basically, you remember I showed you the first slide as a circadian clock, the sort of actogram, this monitoring of sleep and wakefulness. So basically, with this Diego Mazzotti, this colleague, we selected people where we gave them a bracelet that monitor the movement. So if you sleep, you don't move. So you can basically monitor the activity across the 24 hours. So the black peaks are activity, and when there's no peak, it's you stay still basically. And in green, we highlighted the time of the day, which is about when you go to bed. So between 10, 11, and 7 in the morning. And then we mentioned, we look at this lack of activity, because you see that the green boxes, there's very few peaks compared to the others. So we took young adults, older adults, about 60, and very old people, like 80, 85. And we took people that are very old, and they are very healthy. So we took people, we chose a population of people who are like 85, 90. They don't have chronic neurodegenerative problems. They don't have diabetes. They are generally healthy. And when we look at how do they sleep, the synchronization. So this is there are 7 days. The lines are days. You see that they sleep very well, and they go to sleep at the same time, they wake up at the same time. So they are really, the pattern of sleep is perfect. So there is a connection perhaps to the fact they are all healthy and they sleep well. Look at the young people, the health as well, look, is disrupted, because maybe the one who's shifted was the start of the night, so they went out, and you have not a regular pattern. And even an older adult, so a person who's 50 and 60, you don't have this synchronization. And what we have from these patients, we have the blood. So we can do, we already did some transcriptomics, and we can start with the gene, the metabolite, epigenetics. I have some data, but I won't show you everything. So what we are doing now, this was my idea, but in collaboration with many people. We presented this project at the European Union called Climate Age, Circadian Functions and Healthy Aging, a liver link, because we want to study the metabolic output, so we look at the liver. And I'm very happy that we are collaborating with Georgia, so with Nina, but also with Dr. Marina, with Julia. It's not here today, but because the idea of this project is to monitor the sleep of people of different ages, and also if they have metabolic disease, like the liver disease. If they're obese, if they're fat in the liver, and try to understand if there's a connection, and also study the molecular mechanism. So, well, let's hope it gets funded, but I mean, many people are interested. We assembled a strong team, and I think that it's a very hot topic right now. Because I think what can we do to be better, to be healthy? Actually, this is not possible to do it in Georgia, so the fasting, and the caloric restriction. Because if you don't eat much, or today you eat, tomorrow you eat less, or you don't eat, you are telling the brain that it has to synchronize the circadian rhythm. So that's a mechanism. So if you don't give food, the brain will synchronize the clock. And at least in model organism, we know that this can increase longevity. I show you the caloric restriction increases longevity. We also know that caloric restriction synchronizes the clock. So we don't know the mechanism, but it's connected. And then also regular sleep schedule. Nobody of us does this, but it's very important. So going to bed at the same time, more or less, wake at the same time. I always have 7-8 hours sleep. Benjamin Franklin, one of the presidents of the States, early to bed, early to rise makes a man healthy, wealthy, and wise. And yeah, so this advantage of healthy lifestyle. Thank you very much.